Thank you everyone for attending today. Good morning. And I will start saying that with this presentation, I will address the efforts and approaches that my lab is taking to help to understand and help solving the fruit drop issue that we currently have. Now, this is an overview of the talk today. And first, I will address the current fruit drop situation in Florida. Then I will look briefly into what we know historically about mechanisms of fruit abscission and drop in both Valencia and Hamlin varieties in Florida, as this knowledge is actually dictating my program on which approaches we can take to alleviate the problem. And finally, I will show some encouraging results that we are starting to have. So first, let me start saying that fruit drop is a natural process. It is developmental, it is endogenous, and as the fruit matures and enters senescence, fruit drop is a mechanism for seed dispersal in nature, but it may be influenced by other external causes. In fact, as we all know, HLB has exacerbated fruit drop in the last decade. And in this table, published by Dr. Tripti Bashist, we can see a testament of this. In the last couple of years, the problem has been even aggravated more. Now, HLB is one cause of this, but maybe there are some others, and there can be endogenous and exogenous causes. It has been proposed, for instance, loss of root density, altered hormonal balances in the tree and in the fruit, maybe of blooms as well. These two are somehow related, but also there are some exogenous factors such as disease, HLB, fungal diseases, other diseases, and also we are seeing some warmer and drier than normal weather patterns. And these factors actually can interact with the endogenous factors, complicating the whole picture. Now, talking about climatic factors, as I was addressing just a moment ago, this is a picture from a grove in Valencia, Spain, from a couple of weeks ago. There is no HLB there, and there is no canker or other severe diseases in that area. And growers are blaming drought and warm temperatures for this total loss in a grove of Ortanique mandarins that were ready to be harvested. So what we can say here is that we are not alone with regards of unwanted fruit drop. So this is the question that we need to answer. How can we confront and solve this problem? And for that, my program is taking two different approaches. A first one that is what we would call a 30,000 foot view approach that is giving us a whole perspective of the problem. And then with what we are learning, a more boots on the ground approach to come up with practical solutions. And we think, I firmly believe that success in this should combine both types of approaches. So in our first approach, we consider the different fruit developmental physiology of both varieties, Hamlin and Valencia. And as you know, at least this was when we had healthy trees. Hamlin matures and is harvested before the tree blooms again. But in the case of Valencia, the maturation process takes more than 1 year. So final fruit maturation and harvesting usually happens when the tree has a new crop setting and developing for next year. So you have at least two crops on the tree. Now, about 20 years ago, before the HLB era, there was an interest here in Florida in developing a mechanical harvesting system, as many of you may remember. And this system would allow to harvest only mature fruit from Valencia without affecting the new developing fruitlets. And this, to be successful, required the use of a chemical. In this case, we were using CMNP that would specifically promote the loosening of the mature fruit without affecting the newly set crop for next year. Now, looking into the response of both varieties to this compound that I just mentioned, CMNP, we can see how in Hamlin, the fruit detachment force, which is, by the way, the force needed to remove the fruit from the tree, steadily decreased, and we are seeing here the black dots, steadily decreased after the application of the compound. And by the way, a fruit detachment force below 6 means an increased fruit drop. Now, in the case of Valencia, the response to the compound varied during maturation. You can see that again in the black dots over time. But the most interesting thing in this case was what happened naturally. And now I'm talking about the white dots. And you can see a couple of decreases in the fruit detachment force. And these decreases were coincident with blooming and fruit set. So we saw that natural fruit detachment force decreased at these two different moments of fruit development. Now the question is why? Why this happened? And the answer is because there was an abscission signal. Now, this concept of an abscission signal in the tree can integrate really well in a communication theory scheme in which we have a sender, a receiver, a channel that channels the message or the signal, and everything happens in a context. This is a very simple concept, and I ask you to keep this in mind because this will be really helpful down the road during this presentation to understand all the issues that we are having with fruit drop. Now, briefly, what this compound CMNP was doing was to decrease the energy levels in the cells of the mature fruit. As you can see here, you can see a reduction in the ATP content in the cell. And ATP is a sort of energy currency in the cells that inform actually the cells of the general energy status between cells and inside a cell in the fruit. Now, when ATP drops, cellular membranes start to be degraded. And this happens always in all plants. And this degradation occurs because of the action of an enzyme that is called phospholipase. In this case, PLA2. So ATP drops and you have an activation of PLA2. And the ATP drop can be transient. That, as you can see here, later levels of energy recovers in the fruit. but the signal has already been sent. And when you have that, in the same time course, you have a decrease in fruit detachment force. And by 3 days later, you have a fruit detachment force close to 0, so you have fruit drop. Now, going back to our scheme, we know that this energy depletion or energy shortage can be a signal. But what we saw also was that by using some inhibitors of this enzyme that I just mentioned, PLA2, and now you can look into the gray squares here, this plot here, by using this AT aristolochic acid inhibitor, we were able to reduce fruit drop by an 80%. Okay, the fruit drop delay related to energy shortage, we were able to reduce it by an 80% by using this inhibitor of PLA2. But also, and this is the interesting part, it is known that new flushes and developing fruitlets, when they are at marble stage, they are a source of auxins, and auxins are transported to the mature fruit across the tree when they can also activate phospholipases. This is something that is known in several plants, that auxins can also activate PLA2 or phospholipases in general, and we show that in citrus. And when that happens, abscission occurs. And what we saw is that our inhibitor of PLA2 or aristolochic acid, again, look at the gray square plot, that treatment virtually suppressed all the auxin-related fruit drop, pointing out that auxins may be another potential signal for fruit drop or for abscission and fruit drop, but only at very specific times during fruit development. And this is really an important concept. Not everything works at the same level during the whole maturation process of the fruit. So all this integrated really well in a scheme in which an increase in the flux of auxins from green young tissues, flushes and developing fruitlets to mature fruit induced PLA2 activation. But since auxin level at that time was transported to the mature fruit was still high, nothing happened, as auxin per se, we know that inhibits abscission. So this was paradoxical, and at the beginning we didn't understand until we saw that these auxin levels dropped in the mature fruit, and then the PLA2 could act, and then abscission and fruit drop occurred. So we can see that auxins, in this case IAA, and the ratio with other hormones are possible signals too. So we are not talking only about energy or carbohydrate shortage, but there are also some hormonal signals that may play a role here. Now, how can we translate this knowledge to the current situation that we have now in Florida? And this is what we have now. This comes from a Hamlin tree affected by HLB, and you can see that we have a lot of different developmental stages here at the same time in a tree that typically, when it was healthy, had just 1 or 2 at the same time, usually 1. So the question is, how will this variety of developmental stages at the same time in the tree affect the mature fruit? And to answer that, we need to take a very brief look at what citrus maturation is. And citrus maturation is a really complex, but very well endogenously regulated process that results finally in senescence and fruit drop naturally, as I told you at the beginning, for seed dispersal. So we have in the fruit, as it matures, develops and matures, a balance between gibberellin levels and ABA levels that actually dictate the ethylene perception and sensitivity in the peel of the fruit. So we need to have a certain level of ABA accumulation to have a sensitivity to ethylene, and that depends on the gibberellin content of the fruit that needs to drop for this to proceed and the fruit mature. But this, as I told you, can be altered by some other factors external from the mature fruit that are, for instance, auxins and carbohydrates. And to add a new layer of complication, all of that is under the control of different environmental conditions, including disease and altered tree physiology. And all of this takes us back to the communication theory scheme that I just told you a few slides ago. And now we can identify our senders that can be of blooms, developing fruit, flushes. We can consider the tree as the channel, and we have identified several possible signals that would be our message, and our message is to drop. And the receiver is the fruit, and in many communication schemes, the receiver is the most important element of this circuit. And all of this occurs in a context that can be dictated by climatic conditions and disease pressure. And in a situation like this, we can do two things. We can interrupt the message or change the message. And here is what we have now. We can actually tag all these as senders and receivers, all these different stages in the tree. And the question is, how can we control this? And this takes me to the second part of my talk, which is where I am going to show you what we are doing. And we are trying to identify signals so we can interrupt the message, just as I told you. And we are also saying some ways to change the message. And here is how we can interfere the message. This is our communication scheme now. So we have the fruitlets, which are our sender, that send a signal that is auxins, that induce PLA2 in the mature fruit, and then we get abscission. And we know that we can actually suppress that message with, or we think that we can suppress that message with AT, that aristolochic acid, which is the inhibitor of PLA2, and that should suppress abscission. Or we can suppress the message just by interrupting the flux of auxins from the fruitlets to the mature fruit. And we did that, and this is what we got. We measure fruit detachment force before doing the treatments. This was February 1st, this was last year. We have been doing this for a couple of years, and we got the same results every time. So you can see the fruit detachment force was really, really high at that time. Then we had anthesis and fruit set by 15 days later, And then we did our treatments with both the inhibitor of PLA2 and with the transport inhibitor. And you can see when we measured 10 days later that the untreated control had a decrease in fruit detachment force. Remember, below 6, you can have fruit drops. And that's what we were having here. But with all these treatments, the inhibition of PLA2 and the interruption of the flux and the combination of both, we were able to maintain the fruit detachment force. So this fruit didn't drop. And we did another thing. We took the sender out of the equation, and we did that by hand removing fruitlets from the tree. Any off bloom that we saw in that tree, we just remove it. So this was a very nice proof of concept that auxins are really the signal and the fruitlets are the sender. Now, we cannot use, and that's the problem, AT or TIBA, these two compounds in our grows. They are not registered for commercial use as they are toxic. And hand removal of all fruitlets in a grove is impossible to do. So what else we can do? Well, what we can do is the other way. We can actually change the message. And it is known in other plants that auxin biosynthesis and signaling depends on zinc levels. And zinc is also related to phospholipase action, to PLA2 action. As you can see in this scheme, on one hand, it is known that zinc deficiency produces the same kind of lipid peroxidation and membrane disorganization by activation of phospholipases that leads to membrane disorganization and probably abscission. We don't know, but we believe so because the physiological effect is the same. And on the other hand, it is known that supplying zinc to the plants produces detoxification. And also we have seen that it produces better fruit peel integrity and size. And we hypothesized that probably here we could impact abscission and we could prevent abscission. So to test this idea, we perform different treatments, not only with zinc, but also with auxins in the growth, but also on isolated fruit in this in vitro system in which we just supplied the treatments through these pipetors. And by isolating the fruit, we were discarding all external factors that could interact. So we were removing all the context. So the only thing that we had was our new message and the receiver, the fruit. Okay, and what we found in isolated fruits was very interesting because we were able to reduce abscission early in the season when we applied the treatment in June. This was Hamlin. As you can see here, this is the control. We had more abscission than in our treatments, but then in August, we didn't have that effect. The same happened with auxin. So in August, our treatment was not working. And we saw actually the same effect in the field. We had less fruit drop when we applied zinc in June and in October, but we had less. We have more fruit drop in August and less fruit drop both in June and October. Okay, and the same thing happened with auxins. So this strongly suggested us that zinc application to be effective should follow a seasonal pattern. It's not the same to apply the zinc in June than in August, as you can see here. So we still need to understand this to establish this kind of treatments. But now, let me show you what we found with yield. And this is the second year that we did this. The first year we didn't see anything, but this is the second year that we repeat this in the same trees. And as you know, these treatments to work need some time, but it's really encouraging to see that we have done this in Hamlin, also in Valencia, but I'm showing just Hamlin as we have 2 complete seasons, 2 different rootstocks, Swingles and US 942. And we have seen a modest but encouraging increase in both number of fruit and boxes per acre after our zinc treatment. And this is zinc treatment during the whole season. And what we are thinking is that if we can actually focus on those time points that I showed you, June and October, and then do the treatment there, and not during the whole year, probably this yield will be much better. And this is what we want to do in the next season. So in conclusion, we have seen that we can actually modify hormonal balance in the fruit with commercial feasible treatments, as the one that I just showed you, but they have to be applied at the right time, and we need to define that. These treatments in our hands, and we have just this couple of years of data, help to alleviate fruit drop and increase yield. And as I told you, time of application is critical for treatment success. And this must be defined for each variety. For each variety and for each treatment. And it depends greatly on the physiological status of the fruit. And as just I show you, we just started with these approaches. So we have a lot of work to do, but I think we have shown that understanding mechanisms of fruit drop have allowed us to find a path forward. And I believe that expanding our knowledge on these mechanisms will lead us to additional paths for success in alleviating fruit drop. And finally, please let me thank especially my research team, especially my scientific research manager, Tim Gast, my lab technician, Marba Ahmed, my field technician, Maria Antonia Martinez, sorry, and my 2 students, Susmita Gaire and Daniel Boachi, who made most of the work that I presented in the last part of my talk. And this is all that I have for today. Thank you.